Hello everyone, from this video you'll get information about DNA isolation. First, we'll know about what is DNA. DNA is deoxyribonucleic acid is a molecule that contains the genetic code of organisms. This include animals, plants, archaea and bacteria. DNA tells the cells what proteins make. So I think everyone are very eager to know how the DNA is isolated. Now, coming to the introduction of isolation of DNA, DNA isolation is a process of purification of DNA from sample using a combination of physical and chemical methods. The first isolation of DNA was done in 1869 by Frederick Mister. Methods used to isolate DNA are dependent on the sources, age, and size of the sample. Isolation of DNA is needed for genetic analysis, which is used for scientific, medical, or forensic purposes. Scientists use DNA in number of applications such as introduction of DNA into cells and animals or plants or for diagnostic purposes. On the other hand, forensic science needs to recover DNA for identification of individuals, for example, rapists, accidents, paternity determination, and plantar animal identification. Now, from the next slide, we'll learn about the sources. Sources for DNA isolation are very diverse. Basically, it can be isolated from animal, living, or dead organisms. Common sources for DNA isolation include whole blood, hair, sperm, bones, nails, tissues, blood stains, saliva, buccal swabs that is cheek, epithelial cells, urine, paper cards used for sample collection, bacteria, animal tissues or plants. Stored samples can come from archived tissue samples, frozen blood or tissue, exhumed bones or tissues. Exhumed is nothing but to remove a dead body from the ground, especially in order to examine how person died. And ancient human, animal, or plant samples. Going to the next slide, we'll know about the procedure. Isolation of DNA basically consists of four major steps. First one is preparation of cell extract, and the second is purification of DNA from the cell extract, and the third one is concentration of DNA samples, and the fourth one is measurement of purity of DNA concentration. Now we'll discuss one by one. Now, coming to the first one, preparation of cell extract. To extract DNA from a tissue or cells of interest, the cells have to be separated and the cell membranes have to be disrupted. The extraction buffer helps in carrying out these processes. Extraction buffer, also sometimes referred to as the lysis buffer, is a buffer solution used for purpose of breaking open cells. Normally, extraction buffers are at an ionic strength 0.1 to 0.2 molar and pH 7.0 to 8.0 that is considered to be compatible with that found inside the cell. Chemicals such as EDTA, ethylene diamine tetraacetate, which removes magnesium ions that are essential for preserving the overall structure of the cell membrane, and SDS, sodium diol sulfate, which aids in disrupting the cell membranes by removing the lipids of the cell membranes, are included in the extraction buffer. Having lysed the cells, the final step in the preparation of cell extract is removal of insoluble cell debris. Cell debris and partially digested organelles, etc., can be pelleted by centrifugation, leaving the cell extract as a reasonable clear supernatant. Now, coming to purification of DNA from cell extract. In addition to DNA, the cell extract will contain significant quantities of detergents, proteins, salts, and reagents used during cell lysis step and RNA. A variety of procedures can be used to remove these contaminants, leaving the DNA in a pure form. The most commonly used procedures are ethanol precipitation, phenylchloroform extraction, mini column purification. Now coming to ethanol precipitation. Ethanol precipitation usually is done by ice cold ethanol or isopropanol. Since DNA is insoluble in these alcohols, it will aggregate together, giving a palette upon centrifugation. Precipitation of DNA is improved by increasing the ionic strength, usually by adding sodium acetate. Now, coming to the next slide, phenol chloroform extraction. Phenol chloroform extraction in which phenol denatures proteins in the sample. After centrifugation of the sample, denatured proteins stay in the organic phase while aqueous phase containing nucleic acid is mixed with the chloroform that removes phenol residues from solution. Mini column purification that relies on the fact that the nucleic acids may bind, that is adsorption, to the solid phase silica or other depending on the pH and the salt concentration of the buffer. Now coming to the concentration of DNA samples, the most frequently used method of concentration is ethanol precipitation. In the presence of salt and at a temperature of minus 20 degrees centigrade or less, absolute ethanol will efficiently precipitate polymeric nucleic acids. With the concentrated solution of DNA, one can use a glass rod to pull out the adhering DNA strands, while for dilute solutions, precipitated DNA can be collected by centrifugation and redissolving in an appropriate volume of water. Now, in this slide, we observe DNA extraction is shown. Four test tubes were shown. First test tube, cells are lysed using a detergent that disrupts the plasma membrane, and the second test tube indicates the cell contents are treated with protease to destroy protein and RNase to destroy RNA. Coming to the third test tube, cell debris is pelleted in a centrifuge. The supernatant, that is the liquid containing the DNA, is transferred to be clean tube. In the clean tube, the DNA is precipitated with ethanol. It forms viscous strands that can be spooled on a glass rod. And coming to the last one, that is measurement of purity of DNA concentration. DNA concentrations can be accurately measured by UV absorbance spectrometry. The amount of UV radiation absorbed by a solution of DNA is directly proportional to the amount of DNA sample. Usually, absorbance is measured at 260 nanometers. At this wavelength, an absorbance of 1.0 corresponds to 50 μg of double-stranded DNA per ml. With a pure sample of DNA, the ratio of the absorbencies at 260 nanometers and 280 nanometers is in the ratio of 1:8. Ratios of less than 1:8 indicate that the precipitation is contaminated, either with protein or with phenol. Coming to the next slide. In this slide, as we already discussed at starting, the first isolation of DNA is generally credited to Frederick Mister, who in the late 1860s was studying proteins which were discovered a mere 3 decades earlier. Proteins and DNA, what's the relationship? While attempting to investigate the characteristics of protein in puzzles, Mr. noticed that weakly alkaline cellular extracts were neutralized, yielded a precipitate. However, the precipitate did not exhibit any of the known properties of proteins, limited though they were. Through further investigations, he demonstrated that precipitable material originated from nuclei and was present in every cell type he examined. Although he published further his findings in 1869, he made very little inroads into further purification this novel substance, which he termed nuclein. hope everyone enjoyed my video. Thank you for your patience listening and stay safe, stay home.